he's got experience in postdoctoral, in software engineering. He's worked in companies. He's won some nice awards, Outstanding Service Award for IBM in Indianapolis, and he got a Pharma Foundation Researcher Award at the ISMB in 2012 in Long Beach. That's really great. He's also been involved in the bioinformatics core capabilities, including single-cell sequencing in San Antonio. Has an interest in that. We heard her in core facility management, and he's under consideration for the faculty director for the bioinformatics core to work with the management director. Thank you, Victor, for coming, and we look forward to your talk.
Okay. Thank you. Can you guys hear me? Okay, good. So thank you, Brian and June. Giving the opportunity for this position. Glad to be here. It's not first time, but hopefully like more time opportunity in the future. And so yeah, I would like to talk about the research ongoing in the lab, and so my lab actually mainly use integrated experiment and a computational approach to examine the transcription regulation in the genome, of course, application in most of cancer. And a little bit of basic knowledge, I guess most of people you already know, but the chromatin biology. And so we know in the genome, 3 billion DNA, not naked, right? So it's wrapped around the nucleosome and make a dance called chromatin. So, but the basic unit is called nucleosome, which composed of 8 histone units, 2 copy of H2A, H2B, H3, H4, which is 8 protein make is a round shape, and DNA is wrapped around this nucleosome called core nucleosome. And of course, there's another called H1 histone, which link the DNA come in and DNA come out. So definitely there's 9 histone, and the composed is called core nucleosome. And because of protein chemistry and all the nucleosome is not just a round shape, but also there's a tear portrait outside that's called histone tear. Of course, each copy histone has a tear, and there's give the number. Likely, if you study epigenetics, you see a lot of H3, K4, and K27, because right under H3 histone, the number 4 and number 9, number 27, 79 is mostly modified and linked to functional. That's why right now in the field, most people study H3, K4, and because they can modify in different methylation, and then can consequence is result in different functional. So also we know this is chromosome, and this is chromatin, nucleosome core. Because this typical the structure nucleosome, histone, and the DNA, you can because it can modify by different chemical reaction. So we know there's acylation, methylation, ubiquitination, simulation, and the phosphorylation, and combine. Often people call this the histone code. Like, of course, the histone code, you don't really include DNA level. That's why usually now we can change it with, if you include DNA, different methylation. Right now we know DNA also experience different methylation step, right? 5-hydroxyl methylation. And that's why we sometimes call it epigenetic core. So study this epigenetic core as fundamentally to address the principle for chromatin regulation. So, and often, people just study, DNA is one linear fashion, promoter, enhancer, but we know this is not the case, right? So, transcription regulation, not just one dimension, and also 3 dimensions. So, still there's 2 separate theory, still group of people thinking still use linear fashion, still can record the gene. For example, you have the 2K, 3K, 10K away. The enhancer use linear squeeze the model can push the transcription starter targeted gene promoter. But most people accept the model is 3-dimensional, which you have a enhancer element and a protein complex bring to promoter enhancer together and the regular gene is called 3-dimensional model. Of course, my lab, of course, working on this model and intensively. And now current challenge is which I talked about, discuss with individual faculty or core people, single cell level, how to address single cell level chromatin regulation. We know there's many single cell transcriptome there, but not many single cell epigenetic regulation data or research there. So there's a many sequencing technique there. Often we call omics, right? If you see the omics-seq. So there's so many technology over there. And the slides, of course, only list a portion of them and also important of them. And, I started working on ChIP-seq and, way back in 2007 and started, just Illumina as first generation sequencing. And also recently we have a ChIP-AXL, if you notice the ChIP-AXL protocol, and we also have a bit of work in this area. And the MD-cap, a lot of work in my lab, and HISIF, another intense work. Of course, the ChIP drip, MySeq, but I won't go to all the work, right? That's to maybe take more time. But mainly I will focus on some of the work ongoing in my lab. So this slide summarizes pretty much, computational tools and develop in my lab past, I don't know how many years, including my postdoc years. So basically, some is already published and some not. And I can classify there's two major categories in the about two. One is the upstream analysis tool, which is we in sequence core and bioinformatics core nodes, and mostly just direct working on core sequencing data. And so this we have ChIP-seq and MD-cap-seq, ChIP-AXL, HISIF, HISIF, and also their downstream analysis, which is people most also look at the pathway analysis, network analysis, and we also have a bit of work in lab doing this kind of, especially I will talk about a little bit of T-cep and the work in this talk, and also other kind of program. And a couple of slides showing, already online and in our lab website and tools published, most for bioinformatics oriented paper. And this is two papers published at Newcastle Research. One is ChIP-AXL, and this one ChIP-AXL I was going to talk about. I don't have time to talk about this one. I did prepare the slides yesterday, tried to do vision talk, but vision talk did the vision talk a whole hour, so I didn't get time to talk this one. And also visualization too. This already published. This two still under preparation. It's just the tool itself can utilize to implement a really different database. And so for outline for this talk, I'm going to talk about 3 parts. I hope I have enough time to elaborate all this. And part 1 is talk about combinatory epigenetic pattern. And the second one is the genomic interaction, which is HISIF work. And this is the transcription nucleosome and relationship, which is the ChIP-AXL. So this part, we already published a kind of proof of concept paper in bioinformatics to try to demonstrate how hit marker model to decipher the combinatory different histomark or DNA methylation and the regulated pattern, which is kind of called epigenetic code. And this is the overview for T-cep. It's under preparation. And so I won't go to so detail and basically, a bunch of chip-seq data. You're being you're based on the chip-seq being done as a signal and use a Poisson distribution determine the binary actually the code 1010. Then if you have a 7 chip-seq data, then you typically you have a 2 to 7 128 kind of a code, like it's kind of for the binary code from machine digital machine, and then you use a hidden mark model to training to get a combinatorial probability. So there's of course in the field there's some tools already use a hidden mark model and publish, and but we have a different elaborate a little bit more on this difference why we still use hidden mark model, because one, of course, algorithm itself different. The other one is we also consider transcription factor top of just the histone mark, because we found that if you don't consider transcription factor in the beginning of chromatin combination, you miss a lot of information. So that's our talk about this. And that we use one typical data, TCF7L2, to training all this, get some results. Typically, this each competition tool, you need some data to demonstrate. So for this particular tool, we use called TCF7L2, and is a downstream Wien signaling transcription factor. There's many people working on this factor chip, and but not really, because so far they found only these factors involve many diseases, but mostly the colon cancer, diabetes, and but the exact mechanism still largely unknown, right? So we still need working a lot. And this transcription factor actually is very important. It's quite a, I think it's understudied in the field, right? People mostly study, but this is really important factor. So if you look at the data, so we training 5, 6 cancer cell lines that we all have our own chip-seq data performed by Dr. Patrick Faham's lab currently in USC. So we training started with training 25 states. Then we after training 25, we have a second step, because you have a lot of empty states, right? Basically, one state is all bunch of zero. So we remove all these zero states. Eventually, we get 18 states. Then once we have 18 states, we rerun the data again. Then you get a more improved probability. So the probability emission figure looks like that. So basically, you have 18 states. There's different transcription factor, mark, and we also include genus, 5, 6 lines. So if you use a cutoff, or basically use 0.2, 0.3, you basically typically you can infer some biology interesting TF state. So in this case, because like I said, many programs they didn't uniquely consider the transcription factor. That's why we typically split our results in 2 groups. One is TF oriented, the other one is non-TF oriented. That's very important, because we really don't care in this case non-TF state, right? So we want to see how TCF7L2 regulates the epigenetic pattern. So indeed, we found 4 states significantly correlated to this transcription factor. And so if we look at the 4 states, their distribution and the gene expression and the CG content or other genomic feature, then you can derive, say, in conclusion, state 3 is basically the promoter state. If you look at this, heavily distributed in 5 prima. And the state 9 basically is non-combinatorial. Basically everything is there, right? So not much, biology meaning. And if you look at state 11, heavily distributed in gene body. But if you look at the state 11, the mark, indeed the 11 you have a heavily this elongation mark. Is that 36 here? Oh, I didn't. Yes, I think it's here. So definitely 11 states is an enhancer, but is a introgenic enhancer, and state 14 is a distributed distal. So this is a histograph bar shows each state's distribution between 5 cell lines. But we remove 9 because basically 9 is not biologically relevant to this case. Then clearly you see the MC7, Pankin 1, you have enough for distribution, 3 different states. But we try to look at this one because some states you don't really have any gene, so we ignore this. So in the next few slides, we heavily concentrate, analyze, look at these 2 cell lines, how this transcription factor regulate this cancer phenotype. So then we look at gene correlated to this 2 cancer cell line with Go annotation and online a lot of pathway analysis. Indeed, we found in MC7 cell line, the enhancer and the introgenic enhancer gene enriched the MC7 cancer cell line. And then use Pankin 1 the gene correlated, you enrich the Pankin 1 cell line. That's very good. Really demonstrate our 2 found fish out the gene enhancer and distal enhancer and the introgenic enhancer is correlated to their cell type specific. And this shows some common gene, and this shows the gene expression. Indeed, if you look at if you remember this is the promoter, introgenic enhancer, distal enhancer, clearly we see this state 11 is introgenic enhancer expression is the highest, right? So that's ignored by many tools. Also, you have H37, 36 is elongation in introgenic, but not really people use a computational approach to report actually this enhancer mark contribute more transcription activity to the gene expression. So this is a typical, a Go annotation. I won't go to detail to visit the slide. And so then we compare to few there's many tools we admit. Then the typical 2 very big tool, from a lab, chroma HMM, away. Then we found only our tools can find the TCF associate the typical pattern 3, 11, 14. We organize all these two because when they design, they really not consider TCF a transcription factor signal. So they only can, determine some, of course, a lot of chromatin state, but really not the TCF transcription factor specific the chromatin. But of course, we want to do more comparison. This is not enough, right? We want to, besides the state itself, we also want to look at the IOC curve, all the kind of comparison, see how they look at. So we are working on this a little bit more work. Then we, as students, like say, the beauty of a wet lab, then we can validation by ourselves. And we, in these slides, we, this experiment, we did 2 different experiments. One is overexpressed TCF7, one is knockdown TCF7. So both demonstrate if TCF7 effect. The other biologically, we also want to validate compare state 11, which is the introgenic enhancer, and the state 14 is distal enhancer. Indeed, which one is contributing high transcription activity? So if you look at the first slide, it's very simple qPCR, select 9, 10, 11 gene, and indeed all the gene is regulated by TCF7L2, right, or activity. And the B is use a knockdown, and which you compare. I don't know if you guys see clearly. Basically, first one is the control and promoter and knockdown promoter, and then is states, basically states 11, states 3 comparison. So if you look at the highest bar change is introgenic. So if you compare these two, you see clearly if you knock down TCF7/2, the transcription factor, the enhancer, intergenic enhancer states dramatically change. All these, statistical, statistical significant, and with 3 triplicates, it's example. And this is actually overexpressed. I don't know which one is overexpressed. Yeah, this one is overexpressed. So both use Lufisri activity to measure the activity, like basically your instructor, promoter, and indeed both experiments demonstrate in transgenic enhancer indeed contribute to much higher transgenic activity to this factor. So all this experiment looks like a simple, but demonstration our can validate our tool. Also validate the biology. Say indeed, introgenic enhancer actually contribute more high activity. So another study case that did a little bit early is estrogen receptor. We know this transcription factor is very studied. 70% for breast cancer has this alpha positive, and many people working on this. During my career, I have many work related to this. I just show a bit of work with this program. Basically, into estrogen hormone intrigued the receptor and go to nucleus binding and initiate the transcription regulation. And in this study case, I use another collaborator in Huang's lab, and we use 2 cell line. One is MCL7 and MCL7T, which means tamoxifen resistant, with both from our own lab. So this is all ChIP-seq data and did a few years ago. And if you look at we have how many mark? 9 mark, and we have each cell line we have almost 110 million. In that state, that many years ago, 110 million is a lot already. Now just half the land, right? At that time we take like each land only like 4 million. Remember GA1X if you use the DNA core, that only like each land only produces 4 million, 5 million, right? If you remember that time. But now, of course, we take a lot of money to do that. But luckily, we utilize this data to publish work. And so, regardless, we have 110 million each cell line and given the mark. Then also we use same, program algorithm, and in this case we have a DNA methylation. So we did, we found there's both 3 states actually methylation related, right? Or promoter driven. So then we also did the same kind of approach to look at, each state is correlated to genomic feature to see if they correlate a promoter enhancer. And we found that there's 5 states actually related to the estrogen regulation. I won't go to too much detail. And and you look at the simple Go annotation, same, both states one, but they regulate totally different gene. I didn't show the, one diagram, but this set of genes different. Although they both belong to promoter state 1 gene. So this demonstration, and, this program can identify in different cancer type and gene regulated by cell type specific epigenetic regulated gene. To summarize this part, basically, we developed this epigenetic tool and can examine, combinatory associated transcription factor pattern, and also demonstrate use 45 data sets by cancer cell line, and to demonstrate TCF7L2 regulate and use experiment to validate the proof of concept and the tools. So we also identify, epigenetic states in breast cancer and tamoxifen-resistant breast cancer state. So now I'm going to second part. So this is ongoing project. Some is published, some is still collect the data and show a little bit preliminary results. So again, one paper we published more biology oriented on nucleic acid research. It's called HISIF, a little bit different in the field with tags approach. So if you're not quite aware of HISIF, a little bit protocol itself. So basically, you use some kind of cutting enzyme, and there's many different cutting enzymes there, right? Hindus-3, ACO1, or MOBA1, and a lot. But for this particular case, both the paper published in 2009 from Jodak's lab, and they use Hindus-3, ACO1, another one, and use also you cut as long as this enzyme can recognize this sigma, you cut it, and then use biting to label that cutting site. Then you can use another enzyme to ligate it. This is typical easy, right? Everybody, if you do the molecular biology, can do it. The trick part is how to efficiently pull down this part, because you technically you can cut many this kind of piece, like kind of chipstick. You put out lots of random, and then how to fish out this. So, but also another important step that's required, your sequencing has to be pair end, right? Because with pair end, then you can see clearly one chromatin fragment from one side, the other one from this side, right? Then you can use this information enrichment, then figure out where the interaction is, right? Basically, you want to look at this portion, right? Regardless how long, we don't know, right? You need a statistical. So my student, Sean Lan, did a few years ago, and he come up with this strategy, and basically, regardless in the, once we get the data, quality filtered data, and pair end, you have 3 different ligation events. One is called random, and the other one called self-looping. The one interesting one, which actually the biology meaningful, called proximate ligation. So basically, you have actually complex to interact can drag 2 components. So of course, ideally this is promoter enhancer, right? But later on we will show some data. This interaction not necessarily is a promoter enhancer. Can be anywhere. Promoter enhancer, and enhancer, or repressor. So based on this scenario, and he developed another postdoc revised this algorithm. Basically, we call the validated ultrasonic fragment, right? So then we use a kind of expedition maximization, bootstrapping to fish out the possible ligation event, and then eventually we get a significant this set of interaction. If you look at this graph, you can see we all demonstrate 3 different ligation events. This green one is actually significant, not only from different fragments, also from different enzyme cuts. Because if you know how many, of course, when you do the modeling, you need how many actually the enzyme cut site there. You can know where it's the actual background, right? So basically, we modeled this proximal ligation event. So we see this is actually for certain mixture distribution, and then we look at the random ligation. This is follow the beta decay background. So because you have these 2 statistics model, right, based on different set, then you can infer this is actually interaction, right? Real interaction. This is the background. Then you can use statistical model inference to see fish out this event. So of course, much more sophisticated. There's a coding, right? And it's just then we use the test public data. There's many public data here, right there, right, from Beamer's group, Jobdeck group, and other Jessica Jones group. So we all utilize a lot of them. Then represent the different sequencing depth from early stage only 10 million, and now they have billion, right, from base group, beta group. And so we all test them with 5%. This is 5% FDR and shows, of course, different sequencing depths, you get a different enrichment. Then we compare to there's some program. I have to clarify that although we compare to other people, but they are both the HOMER and the HICCUPS, not really actually statistics model the program. So, but they do claim, you can use the HIC data and can, regardless the resolution or what, then you can identify two fragments, actually their interaction. So we utilize these tools, although they are not a statistics model, and we compare. Regardless, we compare the HICCUPS, very successful, and the HOMER also a little bit overlap. Because the difference is the HOMER, the program itself, they require at least 20 KB. So imagine each fragment, sometimes they can go to 40K, interaction 40K. So our, fragment size goes around 2K to maybe 20K, 10K. So because if you look at these slides, this fragment can be variable, right? Depends on the sequencing depth and cutting enzyme efficiency. But the HOMER itself didn't do a good job. Regardless, they have this data. So then also we compare to primary replicates with our tool. We also get a very reproducible our tool, almost 65%. So, but we are also working on with different FDR. See how many. I didn't show this graph. We have this graph, and clearly, you trade off. You have certain FDR and certain overlap. Clearly, there's a balance. Same as ChIP-seq, right? If you work on ChIP-seq, same thing. It depends on p-value, FDR, and you can get different sets of peak. And then, importantly, you need to balance the static value and also biology meaning. Then we also continue to look at the two sample, biology sample. That's also important. Many, investigators want to know not just one condition, also compare different cell line or different condition, right? Mostly it's two different conditions. Then we use called Landau-type kind of distribution, characterizes two different cell type. Clearly, this is MT7, this is Panther1, or this is GM, this is MT7. There's a differentiation, the binding pattern there, right? So size 1, size 2, exact same. Then we use this model roughly approximate cell type specific interaction. Right now, the lab is validated this set of a gene. Say this set only interaction MT7, that set only interaction GM. So we already have started working on this validation. So we definitely need to question us, right? So then we further, test the because all these two, both cell line has a lot of epigenetic data there. So we take advantage of this public data, including ourselves, our own data. Then we look at this is like I mentioned, if you based on the gene structure, right? Promoter 1, promoter 2, and this one promoter, one second gene. See, ideally, most people think it's just one gene in interaction on promoter, right? But reality, we have a lot of data shows promoter 1 can interact with promoter 2, and promoter 1 can interact with promoter distal 2. So then this distribution, of course, majority is PD1, but we also see a lot of PD2 interaction. Then you look at the ex-histo mark. This is a promoter mark. This is an enhancer mark. Indeed, in promoter region, you more enrich this mark, right? In enhancer, you enrich this enhancer mark. So in line with our category. Then we also look at the gene expression. Indeed, you see the promoter enhancer gene expression is much higher than random gene. So all about zero, about 1 actually. So if you know the gene expression, if expressed is larger than 1, right? Log 2 is 0. So this indeed all the enhancer promoter enhancer interaction gene is about 1. So now I talk a little bit another study case. This one is in K562. This is because this is tier 1 encoder cell line. There's many data. Also, there's a HISIF data. That's why we work out this USM program. We identify different chromosomes. This cycles plot, including both introgenic and interchromosome and intrachromosome interaction, right? This is very fancy, and this is actually within chromosome interaction, and that we totally identify around 90,000. Of course, we admit that this data is a little bit noisy because that's first data from job back group, and we likely identify lots of false positives because of the enrichment. But regardless, we're able to from this 90,000 interaction, we correlated to many histone marks. First, we hierarchy classing interaction 1, right? Interaction, you always have 2 sites, fragment 1 or loci 1, fragment 2 or loci 2. And then you first, typically, you just classing 1 side. You got the 12 cluster, 1 is typical entity epigenetic mark, right? That's understandable. There's still a lot of region, no epigenetic annotation there yet. And then we simply correlation to site 1. So you see clearly, if you have this mark in one side, you have another mark in this side. So chromatin actually share the 2 loci share very similar chromatin environment. But of course, not 100%, right? You clearly also see not 100% here. So maybe, like I said, this is false positive or maybe another information there, right? We can't make any conclusion. But regardless, we share very similar chromatin environment on both sides. And this is enrichment test. I won't go to detail. And more interesting, because encoder, there's many transcription factor chip-seq data. So that's all the real chip-seq data. It's not like a transfactor database predictor. And so if this is a clustering 12 cluster, then you correlate it to clustering with 45 chip-seq data in K562. Clearly, you can get the enrichment of certain transcription factor in certain class. Right? That's very beauty. See, clearly for this CTCF red 12, 21, you guys know that this involved a cohesion complex involve chromatin structure also within the one cluster. And we also look at GATA1, GATA2. Where's GATA1, GATA2? Really clearly is active cluster. So this is the transcription factor clustering, and our network is all this from here 45 transcription factor, and some is more down to the class based on the K562 expression. Some is up, and we did get this network use a certain algorithm. So clearly, we are able to program, able to first classify the HISIF to interaction, then correlated to epigenetic mark, then correlated to transcription factor. Then we look at the expression, and also we found there's a 2 typical chromatin linkage, or we sometimes call chromatin harbors, right? Because a bunch of transcription factor or chromatin interaction over there. So basically, if you look at both LOSA1, LOSA2 correlated gene, so like I said, we not necessarily LOSA1, LOSA2 correspond to the same gene, right? If you look at the previous slide, they maybe represent 2 different genes. So that's why gene expression may not be the same. But regardless, LOSA1, LOSA2, the gene expression is high in this cluster, is higher than random. But in this A cluster, its loads are random. So we can call this the repressed cluster. This is the active cluster. So in here, we miss one cluster. Often people talking about is a bivariate, like false. But in another data set, we indeed found that those are one, those are two, not necessarily in same trend, right? So this is what we call a no. But in this data set, we didn't find this other, genes pattern, but they should have, other data maybe have that. So then we further, use validation, simply knock down GATA1, GATA2 to perform the RNA-seq. Right? Ideally, of course, review also asked why don't you knock down to the high C, right? But that time we don't really have this technology in the lab yet. We just computational come up first. So, but we did actually can prove some sort of validation because we fish out after identify GATA1, GATA2 class related gene. This portion gene indeed some involve the looping, but some not. But we also identify some nothing to do with the looping, right? That's also understandable because first we not really got to knock down the high C data, right? Secondly, the high C data itself not really high sequence that's not, right? So both cases, regardless, we sort of validate indeed this GATA2, GATA1 to regulate 3D chromatin regulation. So for I have another case. This is really ongoing in the lab. We just finished generated time course estrogen treated data. This time, of course, we have our own technology already mature, can do right away, and the TCC adapted from Jasmine Zhou's lab. And so we again MC7E2, like I said, my lab did a lot of related actually work. And so we have a 5-time course, time 0 without treated, 0, 1 hour, 4 hours, 16, 24 hour, and also compare tamoxifen resistant cell line. So because we wanted to find what tamoxifen resistant the cell line, what the chromatin structure different, right, from 3D to address the resistant. That's really, I think we are the first one to look at, this particular biology, cancer biology questions. And see, if you look at the low rates, each time we almost get, close to 200 million tail end, right? So imagine, of course, right now sequencing is much cheaper, but regardless, there's still a lot of sequencing there. And total, we got a 1.1 billion tail end rate. But I didn't show another slide data. We actually order 6 data points. We have chip-seq data. We have 7 chip-seq data, CCF, alpha, and 5 epigenetic mark. So all this data we were going to integrate to address from 3D chromatin regulation how to cause this tamoxifen resistance. So this is a little bit of analysis, and so this is still, like I said, a working in progress. And this is, typical task approach, to 2D dimension correlation. And this, if you, use the kind of observe expected, to dilute this, the matrix. And this is typical compartment approach. We use, in this case, we use a typical first paper, science paper. They use called compartment. Basically, compartment A defined as the more active signal, which is commonly supposed more open, and the compartment B is the more repressed, and then is the supposed more condensed chromatin. So this is to replicate the Pearson correlation, and so then further use called PC1 value to construct the countermeasure. This is the chromosome 1 as an example. Clearly see this 5-time point different from time. So if you look at the whole genome, you really don't see too much difference, right? They're all kind of same color, But if you look at zoom in, I should have put this slide before that slide. So then if you look at just the one region, a particular chromosome 1, you see this is 6 points, right, from time 1 to 24. Clearly, in this next size, the region, this is compartment A, compartment starting in 24 hours disappear, but to do tamoxifen resistant to flip. So you imagine here is, so for this case, really, interesting because we can identify this kind of pattern, right? Of course, my postdocs are now working on this. See how many actually this kind of A to B, or A, B, B kind of a pattern there is a kind of a statistical summary. Oh, this is kind of okay. So then we look at this, if you typically look at the compartment number, and you really didn't see too much different, right? Each chromosome, you get a very similar number for compartment structure. That's, really not surprising, because regardless move around, if we have another data we didn't show here, regardless chromosome structure, people already test, or it's very stable. But between different cell, different biological condition, they maybe move around, but the whole structure is still there. So we need, of course, in the field, need more data to support this kind of conclusion or information. So if you also look at this interesting graph, so we simply to graph the number from time point, see only time 1 hour, you see the compartment AB is converged. So the number of compartments the same. So this is there something there? We don't know yet because from biology point, cancer biology point, because indeed many paper, many study include hours. 1 hour when you estrogen treated at 1 hour, the transcription level is the highest change. So which means most of the expression change, transcription binding change, everything, including ER for binding, right? We have data, we're not there yet, but that definitely there's something going on in 1 hour. But after long term, something stabilize. So we really need a time series data to address how to estrogen affect this tamoxifen resistant concept. Okay, so of course this project is still going on. We need a lot of work to do, maybe another half year or more. And so summarize this part, and basically, we develop this computational tool and different from field and use 3D interaction to look at HISIF data, and we use different study case to demonstrate with validation and our current data to try to address more cancer biology or biology-driven hypothesis. So I still have maybe 10 minutes because we started late, and talk about the last part. Also very interesting, the very big part of in the lab, and this is called chip axle. And if you guys notice, and there's just last 2 years maybe, and the paper come from Frank Pugh's lab and chip axle. But what we have is a little bit improved from our collaborator at Ohio State, CCMB lab, and they use called tail end strain specific. So we call chip E10, and so we already published a few paper on biology aspect. More address how to AR estrogen receptor use the chip axle to examine the cancer estrogen castration resistant cancer. But we wanted to also, my postdoc already left, he actually looked at a different angle, use this tool, and basically a little bit of this protocol. Of course, if you do the chip-seq, most of the steps is the same. Okay, your routine cross-link and your sonication. But one step to improve they add one called lambda exonuclease called exo. That enzyme, the function purpose is can eat DNA until the end of the binding border. Right? That's very beautiful because you can typical ChIP-seq you can identify sequence 300 to 500p, right? But this one you actually claim can identify one base resolution. Then further we use strain-specific sequencing protocol. Not only look at also look at the sequences to adapt, both 3' and 5'. Because this information very useful. But original protocol, they didn't really use the two adapter, right? We really improved. But this it turns out this improvement really provide much more information from our aspect. And the algorithm already published in the past. Basically, we have a 4 step. Very quickly, first step is 2 step, very typical like a ChIP-seq, you call the P region. And this utilizes called 3' formation end. Typically, if you forget the ChIP exo, if you use 3' end reads, this just ChIP-seq data. Then if you use a 5' exo reads called R1, then you can use identify the border. So basically, you have first you have a big region like a ChIP-seq. Then use a 5' end, you can identify actually the pick a border, right? Then you know where the motif is there. So use this we use GATA2 or FOX1 to as a study case because we have our own data. So firstly, FOX1, and we know FOX1 is FOX1 and GATA2 is both a pioneer factor and play very important role to regulate the nucleosome, also consequently regulate cancer biology, cancer progression. And but we know FOX1 already well-known pioneer factor in many papers, but GATA2 is still unclear, although there's a paper there. So that's why right now in the lab, we want to further address this GATA2. So if you look at this FOX1 first, in CAP is prostate cancer. So you see the 4 different, we use our program, we can stratify 4 different patterns. So one typical interesting we found called pair end border. So which means 2 border is paired, right? If you look at this case. Yeah, if you look at it here, you actually see the border 2, 1 here, 1 here, paired. But not all the border is paired, okay? So yeah, so we identify, paired, not paired, and the multiple paired. So, but if you look at the paired, very interesting, they distribute like a bimodal, right? So there's a one peak around 8, another peak around 11, 22. So basically, typical, looks like this is a different binding pattern. Looks like, bimodal FOX1, there's a, dimerization to binding 2 strands. So it's interesting going on there. Then we further look at, use biophysical information to further distinguish the model. See, all this clearly show the model binding is affect the motif. Right, so this is the distance, how the motif found close the distance. Luckily, for FOX1 data itself, almost 99% of border we identify there's a FOX1 motif there. So clearly, FOX1 is a direct binding on the motif DNA, right? Not the indirect. Some transcription factor can interact, tether on the transcription factor, right? But this FOX1 clearly just sitting on the DNA, even there's a condenser. That's how pioneer factor is, right? So then we calculate the nucleosome because this clearly pioneer factor is related to nucleosome. So we use public data at that point. Now we have our own data. The public data, and we particularly look at the FOX1 border and the 3 different chromosome a nucleosome, right? We know the chromosome already at the beginning of the talk. And so if you calculate the nucleosome distance, if you look at the 4 different patterns, clearly, they are different. 1, 2, 3, 4. The nucleosome distance is different. This is because he used a certain linear regression to calculate the distance. But of course, we acknowledge at that time the data, the public data itself, not enriched. We only, not the genome-wide look at, we try to do this more. I will talk a little bit next few slides. So again, then further we, because FOX1 is the pioneer factor, can regulate the remodeling. So then we further pick up 8 remodeling factors, data ChIP6. So ideally, we want to do, look at see how these 8 different chromatin remodeler, nucleosome remodeler, how can different correlated 4 patterns. Unfortunately, for this particular data, we didn't see too much difference. Right? Although we found all enriched, the 8 remodeling factor, all heavily enriched this border we identify. But that's not our purpose, right? We want to see how 4 different binding model and correlate certain remodeler. But we didn't find it. But review also question of us. But regardless, we have some data demonstrate our the border at least correct, right? So this is a histone mark also clearly to do. So we need to do more work, try to understand which histone chromatin remodeler should regulate to compete FOX1 or correlate regulate by FOX1 to the job. Maybe this 8 chromatin remodeler we didn't select correct, right? Maybe there's 80 different chromatin remodeler there. So further, like I said, work in progress. We, of course, we have data, we have paper, we want to continue focus on this topic. So now we have a new data on GATA2 and in LINCAP, both treated and treated. But we want to talk to treated and treated first, right? So we have a ChIP-AXL parent data and treated. So data quite enriched, each one we had 200 million, right? Each one. So ChIP-AXL definitely needs this kind of data, right? Not just a simple ChIP-seq data. So this is typical. So then we look at compared to FOX1, indeed we also found GATA2 is have 4 different binding models, exactly the same as FOX1. But if you look at this bimodal, the number, the distance different. The gap is for this is FOX1. Both untreated is 11, 12, because I presented that before. But the GATA2 is different. See, this is on 80 base. This is on 21 base, right? So that's a difference. So we can say we don't have enough data yet. We're still working on that. So FOX1 definitely is the pioneer factor, but GATA2 may not be the clearly pioneer factor function, right? They may be between, right? Use hierarchy model, say FOX1 recruit the GATA2. So also we found actually interesting that, have this slide. So interesting, besides GATA2, we also found that FOX1, the bigger motif there, right? So this is clearly show, so maybe there's a, here we found that besides GATA2, also FOX1 there. But in FOX1, you didn't see the GATA2, right? So there's clearly difference. So we need to address this more. So now we are, we already talked about this slide. So now we are working on called Midden's ChIP-seq. If you guys also do ChIP-seq. So this is different, a little bit for typical ChIP-seq. ChIP-seq, of course, the most common link, right? But before the common link, we also Midden's seq data. But we do the first to do the mononucleosome, Midden's treated, then do the ChIP-seq. So we have a lot of data already almost finished. Ideally, this already done. We have almost 1 billion, 100 millions, 1 billion rates for this the duplicate, and all this data almost finished, maybe end of this year, and all 2 replicates. Total we planning 2 billion for all these samples to 5 billion. So the idea is to try to, I don't know if I have a slide here, to look at so this look at how the nucleosome pioneer factor GATA2 or FOX1 to correlate the nucleosome. Like I said, when we did the computational approach to look at the nucleosome and the FOX1, the data not enriched. Now we have enough data sequence. We almost can identify 12 million nucleosomes. So if you, if your total nucleosome, you calculate whole genome 15 million, but we can identify 12 million. So we pretty sure this is already saturated. Because besides 50 million, just the theoretical number, right? You really don't know how many there. So with this data, with the triple axis data, we definitely can infer some, very interesting biology, see different pioneer factor model, right? One is, binding on the nucleosome, and we also have a H1 chip-seq data, try to see if H1 how to compete with FOX1 or GATA2. So also we look at the nucleosome positioning. We found that there's a 178 peak. So if you do the nucleosome positioning group, this number is very good. I already report average the peak is 178 for one nucleosome distance, because there's a depends on the this variation depends in the promoter region or the enhancer region, condensed region, or open chromatin region. So really, this number is all in line with the reported. So now we have tried to do more, of course, dinucleosome, multiple nucleosome, how they distribute. So this is definitely a project we're going on. We try to shoot a good paper. And so finally, the model, like I said, for all this, we try to propose a model because the nucleosome is dynamic to rotate it, right? Then because the H1 to collect the DNA in and send the DNA out. So, but because the FOX1, the pioneer factor property, they can looking for the DNA motif, right? FOX1 motif among so condensed, then make sure to rotate. If once you found the motif, we can remove, reorganize the nucleosome, make it open, is of course as a nucleosome. ATP model of nucleosome. That's why we wanted to remodel it, right? To look at then binding there, then recruit all different complex come in, then eventually, make the chromatin regulation. That's our model. We want the data to test with the model, a lot of data we try to do this. So for conclusion, and I think this is this last slide. The conclusion, of course, we have this program with chip pair and chip axis data, and try to examine pioneer factor, how they regulate the nucleosome, and result in transcription regulation. And of course, down the line, our particular biology is prostate cancer biology. All the data is the linkage, and we future if we got the funding, we can do the linkage resistance, right? You guys do the prostate cancer, linkage ADL. So we will also do that set of data and can compare how to cause the resistance from nucleosome structure level. So this is another layer. So for conclusion, this is the lab people doing the work, and Zhenqing did Chibasa, he left, now working in the Mayo Clinic. Shanlan, pioneer did the HISIF algorithm. Now he just wrap up a postdoc in Stanford, going on faculty job. He chose to go back to China, so also he has opportunity in America. So and the other people working on HISIF, these two guys working on the chip axis, continuous chip axis, and all collaborating. Xinhua, we are working closely on S3 biology question, and a lot of data come from, and other Xinhua we're working on closely multiple projects, and of course, for Patty says, we're still working on different projects. So I stop here, maybe a little bit already over time. take any questions. Yeah.
Transcription factors. Lots of folks find ChIP-seq to identify transcription factors to be challenging. Do you have some expertise you can share on how to make that work?
I mean, use ChIP-seq build a network.
I'm sorry, using ChIP-seq to get transcription factors. To pull out the transcription factors.
This chip
Has that been challenging or not?
No challenge. I think a ChIP-seq protocol for transcription factor already established. Many encoder group. So you're talking about technical issue or the?
Yes, technical.
Already you have issue there. So. Oh, okay. So my group actually more beyond just the ChIP-seq. We already do the mono-seq, ChIP-seq. So yeah, you guys, we can talk about the technique. We don't have any. So Chairman Wang's lab, Peggy Farm's lab, both they routinely did. If you guys have a technical issue, we can talk about this. To us, we don't have any issue. Yeah. And from the technology, from the analysis, or like this to me is very established. Yeah.
We have thought around histone. So the advantage of using macroglottal nuclease is when you want to look for histone modifications.
Yeah.
For transcription factors, you need to fix
That's
the
why
proteins too.
That's why ChIP-axle. That's why this data when better fits ChIP-axle already you have a border, right? So you can close our algorithm can find a close to nucleus. But if we use a typical just conventional ChIP-seq, you really miss that border. That's why we decide to use first use mononucleosome digest, then use ChIP-seq. It's mostly histone data. Then you look at the see if really this actually is in 147 base resolution, right? Because you already know the last figure already see the distribution mononucleosome. Then simply you look at in each nucleosome if you have a FOX1 or GATA2 ChIP-axle signal. So this is information to us is very powerful because you really can see indeed that this is a regular nucleosome by this factor or not. Right? Then we, of course, we do need more experiment to demonstrate knockdown, whatever, to see how this. But data itself, already very enriched, can show some pattern.
I have another question. I have a biological question with the LNCap work. So I realize that you were probably getting the data and your collaborators are. I noticed there doesn't seem to be much difference between the GATA2, the vehicles versus the dehydroxy testosterone. Are you making sure that the LNCap cells are still responding to androgens because they are known to be notorious to go from androgen responsive to non-responsive with passage, and that's actually why they're a nice model for prostate cancer from going from androgen responsive to irresponsive.
Yes, that's good. So that's why typical, because again, like I said, my research is not really, really typical cancer driven. Also use collaborative, but their lab said, for example, Chipman Wang's lab is, typical from Mark Brown's lab. So they, of course, address most of it. So typically, when you design the experiment, not just link cap, right? First, you have a link cap, you have a DHT treated. Then you look at the AR binding. And in basal condition, for example, non-treated, AR binding very minimal, right? But you do see some ChIP-seq AR there, but not that much. But after treated DHT, you see dramatically increase the AR binding. Same as the ER. So then of course they also compare resistant. Then of course we also have this data, but again we not really address the AR biology. More like a look at the chromatin.
No, but that may explain if you're not seeing much differences. And also you've noted, like you pointed out in a slide, that you have like 2 biological replicates of those experiments. So that may also be. But if you're going from a cell line. So the point of it, the point of exposing LNCAP to the hydroxytestosterone is to look at androgen responsiveness,
Yeah.
or androgenism, like non-responsiveness. But so that's why I was asking: are they looking at in androgen responsive conditions or non-responsive conditions?
Is this, yeah, you're right. We maybe talk about different biology question. In this particular case, we not really want to address estrogen response biology question. We don't want to even say for publication purpose, we even don't want to address estrogen alpha binding, right? Because we want to more address the pioneer factor. So in the estrogen, of course, in LNCAP, not necessarily AI is, of course, AI is a major play, but not necessarily AI is important in, this. We typically, if you look at the chipstick, AI binding is in the nucleosome depleted region. So it's really not much related to this particular topic. Of course, you want to talk cancer biology, that's different story. We do have other paper show DHT treated, right, in AR versus GATA2 FOX1. We have an NAR paper a few years ago from Chapman Wan's lab. We helped the analysis. So we really see the three hierarchy structure. Before treated, you have GATA2, FOX1, AR binding pattern. After treated, you see another binding pattern. There's really three-layer structure. That paper we also show that knockdown GATA2, knockdown FOX1. So yeah, I really, don't do too much cancer biology.
But my point is not related. I mean, because of the model you're using, I think it's an important thing to check.
Yeah.
My point is more related to the GATA2 data that you've put on your slide.
Yeah.
So the point, from my understanding, the point of using the GATA2 data is to show that there is a difference between vehicle versus DHT.
Yeah, you're right.
Okay,
We don't.
So let me just finish, okay? So then, because I want to make sure that, like, I apologize, I didn't explain it properly.
Yeah, okay.
So, and I'm a biologist, systems biologist, so I always go, I have worked with
good,
LNCAP, so that's, I
need
hate
to learn
them.
more biology, cancer biology.
I hate LNCAP cells. They're always ready in the weekends, never
I
in the
thought the
week.
LNCAP
So,
is easy to calculate.
No.
My students have, I don't have experience.
Nah.
Yeah. Okay.
they're always ready in the weekends, never in the
Okay.
week.
Okay.
So you have to come in weekends.
Okay.
But my point was the GATA2 that you've shown, there didn't seem to be much difference.
Yeah.
Vehicle versus DHE. Okay, so it's an overall, right?
Yeah.
However, so to me, there's two things. It's either the treatment's not working and there's no difference, or it's not a GATA2 because there's 4 GATAs in that family and their binding sites can be a bit related. So that was just what I wanted, like where I wanted to go.
No, definitely needs of this point because so this pattern distributed same doesn't mean the same gene. So basically, before treated, you have 5,000 genes, for example. Then after treated, maybe you have 3,000 genes maintain regulation by GATA2. Another 2,000 genes is not by GATA2. That's what we want to look at, right? So, but the interesting, your question, right? Because the interesting is the binding model is same regardless of treated and treated. That's expected, actually. You're right. We want to find more. We don't know yet. So, but at least this data shows the binding PBS model is the same. That's exactly what we try to do. But the other point is antibody, the experiment not work. That's definitely not true because we have a duplicate. GATA2 antibodies established, right? You can't say, people already do many antibodies, because you have a form encoder rule. As long as you have a replicate antibody, this antibody commercial variable, that's considered validate. Then if you still don't recognize, then we can't do anything, right? So basically, I don't think this is 2 point. I appreciate your point, but it's not the case. So antibodies work, experiment definitely work. We did a lot of time, but the model the same interest. That's why we need to dig out exactly. Okay, yeah. More questions? Okay. Thank you. Thank you, guys.